Greetings. Welcome to the Vaxil Bio call. It's now my pleasure to introduce your host, David Gorin, Chairman and CEO. Mr. Gorin, you may begin.
Thanks, Rob, and thanks everybody for joining this morning. Appreciate your time. My name is David Gorin, Chairman and CEO of Vaxil Bio Ltd, and happy to have the opportunity to speak to you about our company. We are an Israeli biotech company. We're focused on novel drug discovery and development, and our platform is signal peptide based. It's a very unique approach, and we deploy those signal peptides to fight infectious diseases and cancer. Before we dive in, we'll just take a second to have the standard forward-looking statement in front. This is not an offer to sell, and from there we will dive in and discuss a little bit about the facts, our history, and some projections and thoughts toward the future. With that in mind, let's start with the high-level overview. We are, as I said, an Israeli-based biotech company. Our product is based on signal peptide technology, one of the few companies that looks through this lens at various diseases. We have active programs in oncology and in infectious disease. Our two lead programs. First, in oncology, we have had a phase 1/2A safety study in multiple myeloma, successful with our product ImMucin. And here we see the impact of what one would expect from signal peptides, which is high immunogenicity, a very robust immune response that is also specific to the tumor cells, so you don't have some of the side effects that you see in many other therapies. We also don't see immune tolerance, and we also bypass the tumor escape. These are problems that you see in other therapies that are emerging now as lead therapies. On the infectious disease side of our business, we had been pursuing a bunch of different areas. We had progressed far along in preclinical programs, including into animal studies in tuberculosis. And based on our general experience, our understanding, and the strength of the signal peptide platform and approach, when the COVID crisis arose and the genomics were announced, we immediately used our proprietary bioinformatics tools to look and see if we could have an answer to COVID-19 with our signal peptide approach. And in fact, within a matter of a couple of days, we found that we did have some good ideas and candidates. We then quickly ran to patent these and map out a pathway toward clinical work. We have a team that is very experienced, both commercially, business-wise, and also in terms of the science. Myself, I've worked in pharma for over 30 years in a variety of roles, commercial, setting up businesses both at Pfizer and AstraZeneca, running them for a while. Also at Pfizer, having led the worldwide strategy business development group, at AstraZeneca, having led the digital health strategy, and having done a variety of consulting in all of these different areas. We have Gadi Levin, who's our CFO, and works on a variety of different startups, including in life sciences. And then we have our two key scientists, Mordechai Applebaum, who heads our R&D program with a strong background in immunology and has been with the company for almost a year and a half. And Riva, who's been with the company for many years and has been involved in our core research programs for a long time, setting up, running, in a very hands-on way. So we're pleased to have this kind of team assembled within the company. In addition to myself and our CFO on the board, we also have two people, Ari Kellen and Sean Langer, who are both physicians, both ex-McKinsey people, as well as Ari having led the Bausch & Lomb US business. We feel brings a strong scientific and commercial presence, and we believe that that's something unique in the way we present ourselves, the way we do our research, and the way we think about products for patients very early on in the process. Which is a good segue into thinking about our products in a pipeline approach. And our pipeline, again, cuts across infectious disease as well as oncology. Our vaccine that we developed for tuberculosis, we ran with a leading U.S. university, and it validated the fact that our vaccine can work in tuberculosis, including in animal studies. And the next step is to secure the way forward for a potential TB vaccine. We've also compared it to the current therapy, the current vaccine, and we see that there could be opportunities there. We've looked also at many other infectious diseases, and as I said, we recently discovered and validated that we have a candidate for COVID. I'll go into those details in a bit to talk about the work we've done and also lay out the path we see going forward. Jumping over to oncology, our lead candidate there, ImMucin, is a MUC1 signal peptide. If you're familiar with some of the oncology landscape, MUC1 is a well-validated, well-understood target. It's used as a diagnostic, and we believe that as a signal peptide, we have the opportunity to change the trajectory of disease there, both in terms of attacking the primary tumor target, but also metastases. We also have additional signal peptide candidates in immune validation ready for preclinical development. We've also in-licensed a polymer from a large university in Israel. This has shown some very good evidence, very encouraging in animal studies for the polymer itself as a treatment, but also for the polymer carrying doxy more directly to the tumor site, making it more effective with the potential for lower side effects. So the combination of all of this gives us a strong presence in a variety of cancer targets. And of course, we also have a mAb program that's a little bit earlier on in its development. And if we think about our company overall, our company is primarily the oncology and infectious disease that we talked about, where most of our resources at the moment are aimed at the COVID-19 program. Our proprietary bioinformatics tool, we call Vaxil, has allowed us to work very quickly to identify signal peptides and mAb that can target various pathogens and cancer targets, oncology targets, that such that, again, as in the case of COVID, we were able to identify possible candidates very quickly. We are a clinical stage immunotherapy, having completed successfully our 1/2A study in multiple myeloma. We got also for multiple myeloma, we had orphan designation both in the US and Europe. We are traded on the Toronto Venture Exchange. And I think from there, if we jump into our patent portfolio, we have almost 40 patents that have been issued or are pending. These patents cover 5 families or 5 general directions, running across vaccines and diagnostics and oncology and anti-infectives very broadly, some and some very specifically. And then our most recent family relates to COVID-19. At this point, I'd like to take a couple minutes just to talk about the actual scientific platform, the signal peptide, and explain where we see our advantage. So signal peptides are rarely used by companies in this particular way. What we do at Vaxil is we exploit the unique properties of signal peptide domains on crucial proteins to develop targeted therapies against cancer targets and infectious disease pathogens. The signal peptides induce a very robust T and B cell response, which is a unique aspect to us. Under normal circumstances, signal peptides don't present on the cell surface, so they're actually, that's why we see a very low side effect profile, a very safe therapy, but also it acts as a neoantigen in tumor cells. And since these neoantigens are not a result of mutation, but are naturally occurring sequences, these sequences will be identical amongst most patients, so across broad populations, and would give us a unique class of therapeutics, a universal neoantigen. The peptide platform targets affected cells, either transformed cancerous cells or infected, by educating or specifically activating the immune system to recognize and specifically attack these cells and only these cells. So in addition, Vaxil's mAb platform also directly recognizes target epitopes and presents us with a whole other set of opportunities to be pursued. So these are why we believe we have unique hypotheses, a unique solution here, where we hypothesize a stronger immune response across a broader population that can persevere over time and even over potential mutations. So with that, I'd like to just jump into COVID and talk a little bit about how we got to this idea. And if you take those advantages that I just mentioned apply them into our bioinformatics approach. We identified these sequences that have high immunogenic potential. What we get is a unique approach with the complete immune response to the adaptive immune response, the cellular and humoral systems. So we see here an opportunity to have an impact that's different from most, if not all, other vaccine approaches that we're hearing about. And in addition, as I mentioned earlier, we're not susceptible to viral escape due to mutations. And therefore, also, we have the potential of not being prone to resurgence due to protein mutation. So we're starting to hear lots of discussions in COVID around whether this mutates, when it might mutate, how, and we'll talk about the second and third waves of the disease. And we hypothesize, because of the platform and the approach and evidence we've seen to date, that our impact can be unique here, potentially in terms of preventing future outbreaks, and even potentially having a therapeutic effect. From there, I'd like to just share a little bit about what we did in our first experiment, about in our first achievements in the first few months before we get to the first experiment. And as I mentioned, we identified the targets with our bioinformatics approach and designed our experiment. We placed orders for our material and have received the material and have begun our research program. So these kinds of steps can normally take many months for companies to achieve, and we managed in a matter of weeks to bring this along and even complete our first experiment. So we've subsequently initiated second experiment and are working on the additional steps required. The first experiment was designed to show immunogenicity, to show that our signal peptides actually prompt a response from the immune system using T-cell proliferation as the metric. We used human donor PBMCs, which were stimulated by peptide-loaded autologous dendritic cells. So these are standard protocols, standard ways of assessing whether a drug will have impact or a vaccine will have impact. And what we saw, fortunately, is that we had a good response, a good immune response across CD3, CD8, and CD4, all of which play different roles here. And the fact that we saw this across all was very encouraging. We learned a lot from this first experiment also about the different peptides that we were bringing to the table, some better than others, various combinations, some things we learned about the manufacturing process. All of that has now been applied to additional manufacturing work that we're doing. And for our next steps, what we anticipate doing is finalizing a relationship regarding infected blood samples so that we have access to patients who have recovered, and then we can test the impact of the peptides on their blood. And that is the beginning of completing our preclinical work, doing our pre-IND and moving into the clinic as quickly as possible. In order to do that, we have to secure the regulatory pathway. No small feat these days. It's a bit of a moving target, but we're already on that and figuring out whether we need to do all of these experiments, whether we can do some in parallel, et cetera. So we will be consulting with regulatory authorities in order to map out that pathway and make sure we understand what would be needed for a clinical trial. In parallel to that, we are in advanced discussions around GMP production. So that would be production in order to do that in that human trial, formulation, and so on, all the work that would be needed. And then, as I said, to have the pre-IND and IND submission in order to initiate that study. While we do that, of course, we're optimizing the work that we do along the way. And that is our story at Vaxil. With that, I would like to just jump over to some questions that have been asked. The one question that was asked was whether we have received government or institutional support for the COVID vaccine. And the answer is no. Not yet at this time. We are in discussions. A lot of the government support is going to players that they already have relationships with, that are already on the landscape, and being a startup, it's a little bit harder to achieve that. But having said that, we have ongoing discussions about that particular issue. Another question about whether suspected problems with impurities and dosage in the peptides have been resolved yet. So the simple answer to that is yes and no. impurities and dosing in a peptide are complex issues. Having said that, we have seen a lot of progress in the last several months on this issue, not just related to COVID, and we have successfully generated GMP production. We have generated proprietary knowledge on peptide production that makes us confident in our ability to do this going forward. If there aren't any other questions, oh, wait one sec, I think there is one more, sorry. So another question of how is our vaccine different from others in development? So I guess from what I've seen of the 30 or so vaccines that are being developed, there isn't anybody else developing a signal peptide-based vaccine. So all of those advantages that I mentioned around our unique positioning with susceptibility with the immune response that we get across the adaptive immune system, our high safety profile, the ability to have impact potentially for future outbreaks, the ability to potentially have impact on the disease itself. I would say in summary, those are the ways that we are unique. So stronger immune response across broader population that can persevere over time, possibly even through mutations. And are there any more questions that people have? If there are no more questions, then I will turn it back over for wrap-up to Rob.
Thank you. This concludes today's webcast. You may disconnect your lines at this time. Thank you for your participation.